Testing. All right. And let's go ahead and jump in. All right, so I believe we left off with the clinical view here of muscular paralysis and neurotoxins. So, tetanus is a condition characterized by spastic paralysis, and it's caused by a toxin produced by the bacterium Clostridium tetani. And this toxin blocks the release of inhibitory neurotransmitters in the spinal cord, which results in an overstimulation of muscles. Vaccination can prevent this life-threatening condition. Yeah, you can get your tetanus shot, and you need a booster every so often. I want to say 7 or 10 years, something like that. Botulism. This is muscular paralysis caused by a toxin from the bacterium Clostridium botulinum, and it prevents the release of acetylcholine at the synaptic knobs. Although the toxin ingestion can be life-threatening, careful injections of it can treat spasticity, for example, due to cerebral palsy, or it can be used for cosmetic purposes to diminish wrinkles. When people get Botox injected, they are getting the toxin from Clostridium botulinum injected into their face. All right, so for the events in muscle relaxation, we see a termination of the nerve signal and acetylcholine release from the motor neuron. That is a termination of acetylcholine release. So when we stop stimulating the nerve, in other words, the nerve fiber, we're going to stop releasing acetylcholine from the synaptic knob. We see hydrolysis, that's breakdown or decomposition of acetylcholine by the enzyme acetylcholinesterase. Again, acetylcholinesterase is the enzyme that breaks down acetylcholine in the synaptic cleft. We see a closure of the acetylcholine receptors. Remember, those are the ion channels that when they bind acetylcholine, they open up and they let sodium and some potassium through. If we are not releasing more acetylcholine and we are breaking down what's in the synaptic cleft, then there's not going to be any acetylcholine left to bind to the receptor and open it up. So the receptor, that channel, will close and the end plate potential will stop. That is, it'll go back to its resting membrane potential. And there's going to be no further action potential generation if we're not stimulating the motor end plate. We see a closure of the calcium channels in the sarcoplasmic reticulum. Remember, the sarcoplasmic reticulum is a calcium reservoir. That's where we store the calcium. And when we are contracting, we release calcium from the sarcoplasmic reticulum. Now that we are no longer causing action potentials, the voltage-gated calcium channels will close. All right, and so we will no longer release calcium. We then return the calcium that's in the sarcoplasm. I'm sorry, yes, in the sarcoplasm to the sarcoplasmic reticulum through calcium pumps. We pump it in to the sarcoplasmic reticulum. And then once we pull calcium out of the sarcoplasm, there's no calcium to bind to troponin, so troponin goes back to its original shape, which moves tropomyosin back in place to block the active sites on actin. It blocks the binding sites on actin. And when that happens, the muscle will go back to its original position due to the elasticity of the muscle. One more thing to say about this. Oh, it's not listed here in the events, but if you go back to cross bridging, remember that in order to release actin, that requires ATP. Okay? So what happens when you die is that the sarcoplasmic reticulum starts to break down and that releases calcium into the sarcoplasm, right? And if there's a bunch of calcium in the sarcoplasm, then it's going to bind to troponin, move the tropomyosin out of the way, the active sites are open, and then we see cross bridges form. We see the myosin heads reach over and form cross bridges. All right. Now, in a normal living cell, there's going to be ATP generated, and that ATP causes the myosin to release the head, right? It causes it to release, I'm sorry, it causes the myosin head to release the actin. All right. But when you're dead, there's not going to be any more ATP. Okay. And so that results in a condition called rigor mortis. All right. It's the breakdown of the sarcoplasmic reticulum releasing calcium into the cell. And then we see a lack of ATP means that the myosin head stays bound to the actin. Now, rigor mortis sets in over the course of about, I want to say about six hours after death. And then it relaxes over the course of the next 12 to 48 hours. So that stiffness of the corpse is not permanent. It will relax over time. And that relaxation occurs not because ATP suddenly showed up, it occurs because the myofibrils start to break down over time. All right, so keep that in mind next time you're watching Forensic Files at home. All right, so the muscle cells, they have only a little bit of ATP in their storage. The stored ATP gets spent after about 5 seconds of intense exertion. Additional ATP is produced through the enzyme myokinase. The phosphate is transferred from one ADP molecule to make another ATP molecule. That would actually result in one ATP and one AMP. But so 3 ways to generate additional ATP in skeletal muscle include creatine phosphate, which you remember holds onto a phosphate group that can get transferred to ADP, making it ATP. Remember, the D is diphosphate, the T is triphosphate. So we add a phosphate, we get ATP back. We can also generate more ATP through glycolysis, although this only produces a net of 2 ATP. Now, if you look, it produces 4 ATP, but we use up 2 to perform glycolysis. So in the end, we're only getting 2 net ATP through glycolysis. But the breakdown products of glycolysis, pyruvate, can go through the citric acid cycle and oxidative phosphorylation, in other words, aerobic cellular respiration, to yield 32 to 36 additional ATP. All right, so creatine phosphate we mentioned contains a high energy bond between the creatine and the phosphate. That phosphate gets transferred to ADP through the enzyme, well, with the help of the enzyme creatine kinase. Um, Anywhere you see the word kinase, by the way, that's going to be an enzyme that transfers a phosphate group. It either adds one to a molecule or takes one away, but it's going to transfer a phosphate group. Any kinase enzyme will. All right, and then this will give us an additional 10 to 15 seconds of energy when we're exercising. All right. A lot of people who exercise take creatine or creatine phosphate as a supplement. And when I started studying anatomy and physiology, I'm old, so I don't know, 10, 15 years ago when I first took it as an undergrad, uh, there was no evidence at that time that the creatine that people took even entered cells. Now it appears that, yes, it does enter cells, but there's not a lot of evidence to show that additional creatine will increase your uh, athletic abilities greatly. For instance, if you load up all of your creatine with phosphate molecules so that it's ready to go the next time you need to work out, it gives you an additional 10 to 15 seconds of energy. Um, so why do people, especially bodybuilders, take creatine? And the reason is it does get taken up by the cells, and then through osmosis, it draws water into the cells, and it makes your muscle cells look bigger. Doesn't actually make them stronger, doesn't actually make them bigger in a permanent sense, but it draws water into the cells to make them look bigger. Again, you produce creatine naturally. Some people take additional creatine as a supplement. All right, glycolysis is Well, lysis means to break down. Glyco refers to sugars, and so this is the process where we break down glucose. Now, when we get to the digestive unit, you'll see that we can absorb monosaccharides like, fructose and galactose, but you actually can't break them down through glycolysis. The liver has to convert fructose and galactose into glucose before it can go through glycolysis. All right, so once you got your glucose, whether you took that in the form of glucose or had to convert it in the liver, you can break it down through a process called glycolysis. It does not require oxygen, and so it's sometimes called anaerobic respiration. Anaerobic means without oxygen. So the glucose can be broken down, and didn't mention, of course, your muscle also has glycogen stores. Glycogen is the branched storage form of glucose. It's a complex carbohydrate. Both the liver and muscle have glycogen. When your blood sugar is low, the liver will break down glycogen to raise your blood sugar. Muscle, on the other hand, is selfish and will not share its glycogen with other cells. So the muscle stores its own glycogen and only breaks it down when you are exercising, when it needs extra glucose in the cell. Anyway, that's background information. The glucose gets converted to two pyruvate molecules through a series of reactions. Technically, you generate 4 ATP through glycolysis, but 2 get used up to perform glycolysis. So in the end, you get 2 net ATP, right? If you make 4 but use up 2, in the end, you really get 2 ATP glucose molecule. I'm sorry, 2 per glucose molecule. This occurs in the cytosol. Now, the last step in glycolysis produces a molecule called pyruvate, and you'll actually make 2 pyruvate molecules. And that's important because pyruvate can then go through aerobic cellular respiration. Basically, the waste product of glycolysis, pyruvate, can then be put through aerobic respiration, but only if we have oxygen. It requires oxygen. Now, this occurs in the mitochondria. The pyruvate gets oxidized, we release carbon dioxide, and so because aerobic respiration produces carbon dioxide, our cells, specifically the mitochondria in our cells, are the source of carbon dioxide in the body. All right, so we transfer energy to NADH and FADH2, and they're going to transfer, those hydrogens, the H and NADH and the H2 and FADH2, those are going to get pumped across the inner mitochondrial membrane, and then they're going to travel through an enzyme called ATP synthase, and it's going to, as you might guess from the name, the enzyme's going to produce ATP using the hydrogen ion concentration gradient. So in the end, this produces about a net of 30 ATP. On paper, it should produce more, but it's not a it's not a perfect process. So I mentioned that the hydrogen ion concentration is used to power ATP synthase, that enzyme, and there's actually a little bit of hydrogen ion leak, which makes it less efficient than it would be optimal. All right, so triglycerides can also be used as fuel to produce ATP, and so a triglyceride is a glycerol molecule and 3 fatty acids for a tail. And you can separate those so that you get a glycerol molecule, and that can be oxidized, but you also have the fatty acids, which have a carbon hydrogen backbone, basically a chain of carbons for a backbone. And pyruvate is a 3-carbon molecule that gets converted to a 2-carbon molecule before entering aerobic respiration, before entering the citric acid cycle. When I say 2-carbon molecule, I mean it has 2 carbons bound to each other. We do something similar with the fatty acid tails. We can actually lop off 2 carbons at a time and feed them into the citric acid cycle the exact same way. And so that's how fatty acids enter. Aerobic respiration. Oxygen is required to break down fatty acids. All right, so here we have a little cartoon. You can see glucose, fatty acids, amino acids entering the muscle cell. Of course, you can also break down amino acids, although the biochemistry for that's quite a bit slower. It yields the same amount of energy as glucose, but in general, the body would just prefer not to break down amino acids, and so the reactions take place more slowly, and uh, the the byproducts at the end are more toxic, and so it's just best not to use amino acids for energy. It's better to use glucose and also fatty acids if you have to. All right. All right, so the glucose comes in, and you can see in the diagram here it goes through glycolysis to produce 2 ATP and 2 pyruvate molecules that travel into the mitochondria to undergo the citric acid cycle. And the citric acid cycle is a complex set of biochemical reactions um, that at the end, basically, we're going to be uh, powering a series of reactions in the inner mitochondrial membrane that pump hydrogen ions into this space between the outer membrane and the inner membrane. And then we're going to use that hydrogen ion concentration gradient to power ATP synthase, the enzyme that creates ATP. Now, so when we talk about it, ideally we talk about it occurring with glucose enters and goes through glycolysis. We can also take glycogen and break it down through a process called glycogenolysis. And this name, again, lysis means to split or break down. Glycogen is the complex storage form of glucose. Lots of glucose molecules are bound to each other in glycogen. And we just start breaking down glycogen by releasing glucose molecules from it. And that becomes free glucose that can go through glycolysis. We also get fatty acids and amino acids which enter, and they can get converted to 2 carbon molecules which also enter the mitochondria and directly enter the citric acid cycle and then power this whole oxidative phosphorylation process. Oxidative phosphorylation, I've used that term a number of times, and basically it's the series of steps that take place in the inner mitochondrial membrane to power ATP synthase to produce ATP. Requires oxygen though. It's also known as the electron transport chain. All right. And then one more source of energy over here, you can see creatine phosphate. We got the P for phosphate. And you take an ADP, adenosine diphosphate, and then creatine kinase will basically just take the phosphate off of creatine phosphate and add it to ADP to make ATP, and then we've got creatine left over. This is during exercise. When the muscle is resting, this image down below, you can see the opposite reaction occurring. We have ATP, and we're going to use this phosphate to charge creatine and produce creatine phosphate. Now we have an ADP, but we're at rest, and so we can go through this process and make more ATP. Creatine phosphate's kind of like a storage form of ATP. ATP usually gets used up pretty quickly. All right. Now, when you go through glycolysis and you don't have oxygen to carry out the reactions in the mitochondria, what's going to happen is that pyruvate has to get converted, and it's going to get converted to lactic acid. Okay, and so lactate, also known as lactic acid, is going to accumulate in the cells, and that lactic acid can be fermented for additional energy. However, its accumulation is going to result in cramping. Okay, and so when you are exercising and you're not getting enough oxygen to the muscle, you will accumulate that lactic acid, which leads to the cramping that you feel in your side. They call that a side stitch. All right, so Energy supply and the varying intensities of exercise. So for a 50-minute sprint, which usually takes less than 10 seconds, the ATP is supplied primarily by phosphate transfer, basically from creatine to ADP to form ATP. For a 400-meter sprint, which is less than a minute, the ATP supplied will come from glycolysis after the first few seconds. Again, initially, transfer of phosphate from creatine, then glycolysis will take over and produce the ATP that you need. Now, beyond that, for a 1500-meter run, which is more than a minute, or if you were to continue running, say in a 5K, 10K, half marathon, marathon, etc. Um, the ATP would be supplied through aerobic processes after the first minute. So you would then send pyruvate through the citric acid cycle. You would also, after several minutes, begin breaking down fat for energy. Now, one of the reasons it's so hard to lose weight is because fat is the storage form of energy, and the cells don't like to give it up. They want to hold on to it long term. All right. And so you can see a little cartoon of that here. For the first 5 to 6 seconds, you've got the ATP that's already present in the cell, and then when that gets used up, it gets recharged by the creatine phosphate. All right, for 50 to 60 seconds of running, again, you get glycolysis producing short-term energy without oxygen. And beyond that, you're going to require oxygen to go through oxidative phosphorylation or the electron transport chain and generate much more ATP. Okay, and you can compare those, right? 2 net ATP for glycolysis, at least 30 for aerobic respiration. That's a much bigger difference. That's a 15-fold increase in ATP production, right? All right, so oxygen debt is the amount of additional oxygen that's needed after exercise to restore pre-exercise conditions. It's the oxygen you owe to your body. All right, so additional oxygen is required to replace oxygen on the hemoglobin and the myoglobin. Remember, myoglobin is a protein related to hemoglobin, but it lives in, well, it doesn't live, it exists in muscle cells and holds on to oxygen so that you'll have a store of oxygen for short term oxidative phosphorylation. Additional oxygen is used to replenish glycogen, as well as replenish the ATP and the creatine phosphate, and then convert lactic acid back to glucose. So skeletal muscle fibers are classified based on their type of contraction and their means of supplying ATP. So for the type of contraction that is generated, we see differences in power, speed, and duration of contraction. Power is related to the diameter of the muscle fiber. Speed and duration are related to the type of myosin ATPase, as well as the quickness of action potential propagation, how quickly we spread that signal along the membrane, carry it along the membrane, and the quickness of calcium release and reuptake by the sarcoplasmic reticulum, all right, which would in turn be decided by the number of calcium channels. So fast twitch fibers are more powerful and they have quicker but briefer contractions than slow twitch fibers. All right, now we can also classify them based on their means for supplying ATP. So you have oxidative and glycolytic fibers. Oxidative fibers are fatigue resistant. They use aerobic cellular respiration. In other words, so I mean, both cells are going to use aerobic respiration, but oxidative fibers are going to have much more mitochondria, which makes them much better at oxidative phosphorylation and the replenishment of ATP. So they use aerobic cellular respiration, the oxidative fibers do. They have extensive capillaries for blood supply to bring oxygen to the cells. They have many mitochondria, and they have a large supply of myoglobin, which of course stores oxygen until it's needed. These are the red fibers in muscle. Glycolytic fibers use anaerobic cellular respiration. They have fewer capillaries, less blood, supply less oxygen, fewer mitochondria, and a small supply of myoglobin, but they have large glycogen reserves. All right, and these are the white fibers in muscle. Now, uh, one way to remember this is that oxidative fibers are going to use a lot more oxygen than glycolytic fibers, and it's the protein myoglobin that makes muscle red. And so the oxidative fibers are going to have a higher concentration of myoglobin, and therefore they will be the red fibers. The white fibers, or comparatively white fibers, will have less glycogen, they will use less oxygen, they will have fewer mitochondria, which rely on the oxygen. All right, so the three types of skeletal muscle fibers are slow oxidative fibers, type 1. The contractions are going to be slow and less powerful. They're used for high endurance since ATP is supplied aerobically, and they're about half the diameter of other fibers, but they're red in color due to the myoglobin, oxidative red. Fast oxidative fibers. Um, these are the type 2A or intermediate fibers. Here the contractions are fast and powerful. They are primarily aerobic, but they deliver oxygen. They have less oxygen delivered to them, a less rich blood supply. They are intermediate in size and light red in color. And then you have the fast glycolytic fibers. These are the type 2B or fast anaerobic fibers. The contractions are fast and powerful, but they're brief because ATP production is primarily anaerobic in these fibers. In other words, they are primarily using glycolysis, which is fast but doesn't produce a lot of ATP, so you can't keep it up for very long. They have the largest size and they're white in color because they lack, well, they don't lack myoglobin, but there's not a lot of myoglobin there. All right, so a single muscle is going to contain a mixture of these fiber types, and there are variations in the proportions of fiber types in different muscle groups. Basically, different muscles will have different ratios of type 1 to type 2A to type 2B fibers. So hand muscles have a high percentage of fast glycolytic fibers for quickness. Back muscles have a high percentage of slow oxidative fibers because they have to continually maintain postural support. Long distance runners tend to have a higher proportion of slow oxidative fibers in their legs. And sprinters are going to have a high percentage of fast glycolytic fibers because sprinters need that explosive burst of energy, but they don't have to go very far. They don't have to keep it up very long. Long distance runners who carry on their running for hours, a very fit runner, a very, very fit runner might complete a marathon in 3 or 4 hours. The top athletes are doing it in, I think, in just under 3 hours now. All right, so sprinters, they'll run for seconds to minutes. So, and this is going to be determined primarily by genes. Your ratio of slow oxidative to fast glycolytic fibers, but it can also be determined by training. And so you can actually develop more type 2A fibers or more slow oxidative fibers if you need to. Through training and exercise. Now, determined primarily by genes. Okay, so there are a lot of genetic tests out there today, and most of them should be treated like they're just for fun, partially because the science just isn't there to say that our understanding of genes and DNA is determinative. But if you have some excess money and you don't care about your privacy very much, there's some fun tests out there. Of course, there are the ancestry tests, but there are also actually fitness DNA tests you can take now, which tell you things like your genetic predisposition for having one type of muscle fiber versus another. The only other way to tell how much of each muscle fiber type you have might be to cut open the muscle, and nobody's going to do that, right? So the genetic test is one way to determine, and they can actually help you determine which sports you may be more suited for. All right, so you can see here we have the fast glycolytic fibers, the slow oxidative, and then we have the fast oxidative, which are going to be this kind of pink-ish color here. All right, slow oxidative, the dark red. All right, okay. So muscle tension. Tension is the force generated when a muscle is stimulated to contract. And lab experiments can measure the tension and graph it. And so until recently, I worked in a lab that actually had this machine, and they would do this uh, with mouse muscles and rings made up of cardiac myocytes to test the tension generated in the muscle under various conditions. Anyway, when the electrode stimulates the muscle electrically, it's going to contract or shorten, and the shortening can be measured on a computer here. All right, so this is known as a myogram. This graph or this recording of contraction is known as a myogram. And you can see the stimulus occurs before contraction. And of course, we know why that is, right? The action potential has to travel the length of the muscle fiber before we can, and then we have to release calcium from the sarcoplasmic reticulum, and it has to diffuse out to the troponin to move it out of the way, to move the tropomyosin out of the way, so that finally the myosin head can bind to the actin and begin contraction. All right, and so there's a period of time between the arrival of the stimulus and the beginning of contraction, and that's known as the latent period. The latent period exists in the lab. It also exists in nature. All right, now this is measured in milliseconds, so it's not very long, but it's there. Contraction does not begin the second we stimulate the muscle. We still need the action potential to travel along the sarcolemma. We need the sarcoplasmic reticulum to release calcium, and then we need it to bind the troponin and move the tropomyosin before contraction can begin. We then have the contraction period where tension increases as more and more myofibrils are going to be contracting. We can also increase the number of myofibers, individual muscle cells in the muscle over time that are contracting. And then after the peak, we have the relaxation period where we are taking calcium up out of the sarcoplasm and returning it to the sarcoplasmic reticulum uh, after the action potentials and the stimulation have stopped. All right, so for muscle twitch, a twitch is a brief contraction to a single stimulus. And the minimum voltage that triggers a twitch is called the threshold. So for the periods of the twitch, we mentioned, the latent period, the contraction period, and the relaxation period. And there's going to be no change in tension during the latent period because we haven't started contraction yet. Then during contraction, we'll see tension increase. All right. And then during the relaxation period, we're going to be letting go. We're going to stop forming cross bridges, and then we'll suck the calcium back up into the sarcoplasmic reticulum. All right, so for motor unit recruitment, muscle is stimulated repeatedly, and as the voltage increases, more units are recruited to contract. Recruitment is also called multiple motor unit summation, and recruitment is another way we could say this is that as we increase the stimulus to the muscle, more motor units are going to contract. More motor units are going to be stimulated to contract. And so remember, a motor unit is the motor neuron and any of the muscle fibers that it innervates. And so one motor neuron is going to innervate or stimulate, a handful of muscle fibers. All right, but that's not all the muscle fibers. And if you had more muscle fibers contracting, you would get more tension or more force. And so we can recruit another motor unit, that is another motor neuron, and its muscle fibers to contract as well. That'll increase the tension, increase the force. All right. So the example here is it only takes a few motor units to lift a pencil, but if you want to lift a suitcase, that's going to require many more motor units. We mentioned before that the muscles controlling the hand tend to have very few muscle fibers per motor unit. And in muscles like the leg, where we're always going to be lifting a lot of weight, those motor units tend to have lots of muscle fibers. All right. So the same idea here, lifting something light like a pencil will only require a few motor units, and then lifting a suitcase will require many motor units, multiple motor units. So above a certain voltage, all units are recruited. So maximum contraction occurs regardless of how much higher the voltage is. Meaning, in other words, you can increase the stimulus, but once you've reached the maximum number of motor units that can contract, then you can't continue with recruitment because there's nobody left to recruit. Recruitment order is based on the size of the motor unit. So the smallest motor units are recruited first and the largest last. All right, so here you can see the myogram at the top. As we increase the voltage, we are going to increase the muscle tension and thereby increase the, well, not thereby, we're going to increase the number of motor units which are recruited and thereby increase the tension. All right. Sorry, I got the relationship backwards there. Basically, the more motor units you recruit, the greater the tension. All right, but you can see here we reach a maximum tension, right? And here we have recruited all of the motor units. There are no more to recruit. And so as we continue to increase the stimulus, we're not going to see an increase in muscle tension because all of the motor units are recruited. And that is sort of borne out by this cartoon down here. You can see at the beginning, no motor units are recruited, and then as we increase the voltage, we increase the number of motor units recruited until all of them are recruited, and we'll see no additional recruitment because they've all been recruited. All right. All right. So wave summation is also known as temporal summation. If the stimulus frequency is set at about 20 per second, then relaxation is not completed between the muscle twitches, and we see the contractile forces add up to produce higher tension. Incomplete tetany and tetany. So this is when, if the frequency is increased further, on the myogram we see incomplete tetany, and that's where the tension increases and the twitches will partially fuse. If the frequency is increased further still to about 40 to 50 per second, the myogram exhibits tetany, which is sustained contraction. All right. So the tension trace is going to be a smooth line without relaxation. High frequency stimuli lead to fatigue and then decreased tension productivity. So here we can see if the stimulus comes 10 stimuli per second, right? Then the waves of muscle tension are going to be separate from one another and they're going to be identical. We've had time to twitch, we've had time to contract, and then we've had time to relax. All right, so they are identical. If you look over on the right, you can see wave summation, where the frequency of stimulus comes often enough that we haven't had time to relax yet. We are already partially contracted when the next arrives, and so we will increase muscle tension that way. We then see incomplete tetany, where the waves have added together to the point where we are virtually constantly contracted. And then tetany is where the waves have fully fused and we just see a straight line on the myogram, and basically that's where we are sustaining contraction and never relaxing until eventually we can't maintain that anymore. The tension falls off as the muscle fatigues. All right. So muscle tone is the resting tension in a muscle, and it's generated by the involuntary nervous stimulation of muscle. Some motor units are stimulated randomly at any time. They change continuously so that motor units don't get fatigued. they do not generate enough tension for movement, and this muscle tone will decrease during sleep. Isometric contraction and isotonic contraction are two different types of contraction. And in short, in an isometric contraction, we're going to see that although tension increases, it's not sufficient to overcome the resistance. In other words, the muscle length stays the same, the muscle does not shorten. All right. So if you are holding a weight while your arm doesn't move, if you are holding a barbell at your side, or if you're holding a weight to your chest. All right. And then isotonic contraction is where the muscle tension does overcome the resistance and we do see movement. So the tone stays constant, but the muscle length changes. You then have concentric contractions where the muscle shortens as it contracts. And then you have eccentric contraction where the muscle actually lengthens as it contracts. All right. So here you can see a gentleman holding his baby. As he holds the baby to his chest, we have muscle tone, or we have contraction in the biceps brachii muscle here, but the muscle itself isn't shortening. There's tension, but the muscle isn't shortening. It isn't overcoming the load. So he's able to hold the baby in place. There's no movement generated. The word iso means the same, and then metric means to measure. And so isometric means same measurement, in other words, no change in length. Now for isotonic contraction, over here, as he lifts the baby to him, the muscle is shortening, and that is a concentric contraction. And then as he lowers the baby down, the muscle is lengthening, and that is an eccentric contraction. All right. With isotonic contractions, we do see a change in length. All right. So isometric contraction and the increase in blood pressure. We see a sustained isometric contractions are associated with an increase in blood pressure, which may be a concern for those with baseline high blood pressure. So if you are shoveling snow, generally the peripheral constriction, that is the constriction of blood vessels in the skin due to the cold, is going to elevate your blood pressure, right? Basically, when you constrict those blood vessels, it's going to force more blood back into the other blood vessels, which raises blood pressure. The isometric contractions of shoveling will also increase blood pressure. All right, so the tension of a muscle, the tension that a muscle produces will depend on its length at the time of stimulation. So the fiber at a resting length generates maximum contractile force. All right. And then we see an optimal overlap of thick and thin filaments. But a fiber that's been shortened, it generates a weaker force. And we see the filaments move less. They're already close to the Z-disc. There's less room for them to shorten, and so they can generate less tension. All right. Then the fiber at an extended length generates a weaker force, and we'll see minimal thick and thin filament overlap or cross-bridge formation. All right. Okay, so here you see the length tension curve. There's an optimal length, that's the resting length there, of about 2 to 2.5 micrometers. All right. Sarcomeres that are already shortened in advance or already contracted are going to be able to generate less tension. And then sarcomeres that are overstretched at the other end are going to also generate less tension. right, and welcome back. Sorry about that. Let me get set up. You can probably tell that much like the gentleman in the picture, I was holding my baby to my chest, and she suddenly let me know she was ready for a nap. All right, let's go back. Live screen. All right, good. So muscle fatigue, yeah, that's the reduced ability to produce muscle tension. Fatigue is another word for exhaustion or tiredness, right? So it's primarily caused by a decrease in the glycogen stores during prolonged exercise. Other causes for fatigue include excitation at the neuromuscular junction. So if there's insufficient calcium to enter the synaptic knob, then we're not going to be able to release synaptic vesicles, right? Remember, the calcium that enters the synaptic knob enters through voltage-gated calcium channels, and we can change the voltage in the membrane and open up those voltage-gated calcium channels on the neuron side of the synapse. But if there's insufficient calcium in the extracellular fluid, then we can't have that calcium enter the synaptic knob, bind to the proteins on the synaptic vesicles, and then fuse with the membrane to dump their contents out. Then there's excitation contraction coupling, where altered ion concentrations impair the action potential conduction, and calcium release from the sarcoplasmic reticulum. In other words, if we have a deficiency in sodium or potassium and are unable to initiate an end plate potential, or if we're unable to initiate an action potential, or if there's a deficiency of calcium and we're unable to release calcium from the sarcoplasmic reticulum into the sarcoplasm. And then cross-bridge cycling. So excessive phosphate, by the way, this symbol PI, the I stands for inorganic. So this is inorganic phosphate, which as far as we're concerned is just plain old garden variety phosphate. You see PI, you can just go ahead and say phosphate. Anyway, excessive phosphate will slow the release of phosphate from the myosin heads. And then less calcium available for the troponin means that calcium is going to be bound to phosphate. All right, so the effects of exercise. We see that changes in muscle. We see changes in muscle from a sustained exercise program. Obviously, we know that. We're going to change muscle size and shape. Well, not quite shape, but muscle size for sure, as well as our endurance. So endurance exercise leads to better ATP production. We are actually going to build more mitochondria. Technically, the mitochondria build themselves. The mitochondria are going to divide, and we're going to have more of them when we perform endurance exercises. All right, and so you can convert fibers to oxidative from glycolytic by producing more mitochondria. We also see that resistance exercise leads to hypertrophy. Again, hyper means above or higher, and trophy is size. So hypertrophy means an increase in size in the muscle fibers. This is due to an increase in the synthesis of contractile proteins. The muscle also increases glycogen reserves in mitochondria, which will increase the size of the cell. More glycogen means more water in the cell. And then it also appears to stimulate limited growth of hyperplasia. This is an increase in the number of fibers. Now, I should walk this back just a little bit. A lot of textbooks will actually say that this doesn't happen, and this may be a relatively recent discovery, and that's why it's not in all of the textbooks yet. There's a cytokine called interleukin-6, which stimulates a type of stem cell called satellite cells to move in to the area and cause those satellite cells to not only migrate to the muscle, but differentiate into a muscle fiber, become a muscle fiber. Actually, it would take a lot of cells to build a muscle fiber, but not just the one. But anyway, so this appears that it can happen, although many textbooks, especially older ones, will tell you it doesn't. However, what we know for sure is that although it may happen, Although we may increase the number of muscle fibers, um, that's not the primary reason that muscles grow in size. The primary reason that muscles grow in size is this first reason up here is because we are increasing the number of myofibrils in the muscle. All right. So when you work out for 2 months, 4 months, 6 months, and you see the size of your muscles increase, one cause of that just straight away might be that you've burned off some fat, which reveals the muscle. But of course, it will increase in size also. And primarily that's because the muscle cells themselves have increased in size. And they do that primarily by increasing the number of myofibrils per cell, per muscle cell. All right, so you can also see changes in muscle from a lack of exercise. This is known as atrophy, where the muscles decrease in size due to the lack of use. And so what the example here is someone wearing a cast. That's called disuse atrophy. Disuse is where you are not using it. And in this case, it's initially reversible, but it can become permanent. It's initially reversible because the muscle is only decreased in size because we have broken down the myofibrils and we've broken them down either to use the amino acids in other cells or we've broken them down for energy. In fact, muscle cells are very hungry. They're very expensive. Metabolically, they require a lot of energy just to maintain muscle. So, when you disuse muscle, you're pretty quickly going to, when you stop using muscle, you get stuck in bed for a month or two. You're going to pretty quickly stop using, uh, well, when you disuse the muscle, you're going to pretty quickly start breaking down the myofibrils um, for energy or for use elsewhere. Now that's reversible. You can always build new myofibrils with exercise and in severe cases, uh, therapy, physical therapy. It becomes permanent over time though because muscle fibers, if they're disused, can be replaced with um, connective tissue, like broad, thick fibers made of connective tissue. And if that's the case, you're never going to convert that back to muscle. Um, it's always going to be connective tissue. There's another type of atrophy called denervation atrophy, and that's where the nerve that innervates a muscle has been severed. And just with current surgical techniques, it's not really usually possible to reinnervate the muscle, and it will atrophy due to never being stimulated, which is similar to disuse atrophy, and it will typically be permanent. You could maybe save the muscle by regularly stimulating it with electricity, forcing it to contract, but uh, there wouldn't be much point if you were never going to regain control of the muscle with your nervous system. All right, so loss of muscle mass with age. We see a slow loss begin in a person's mid-30s, but this is largely because they are less active. I'm in my mid-30s. I've got 3 kids, 2 in diapers, and it's true. Every time one's born, we do spend about 6 months sitting on the couch, because babies don't want to get up and do stuff. They want to lay there, and but then they get bigger and you start playing with them. I'd like to argue against this statement just a little bit. You play with them when they get bigger, and then you can lift them over your head, and that's exercise. You see a decreased size in muscle, power, and endurance regarding skeletal muscle. You see a loss in the fiber number and diameter. Just over time, muscle fibers get damaged and die off, and they aren't easily replaced. We can develop new muscle fibers through hyperplasia, as we mentioned before. I say probably because not everyone's in agreement on that yet. But you are not going to replace all of the muscle that you lose over time. You will lose muscle at a faster rate than you can replace it. That's for sure. So we lose fibers in number. They also become smaller as we lose myofibrils. We see decreased oxygen storage capacity, less production of myoglobin. We see a decreased circulatory supply to the muscles. All right. So the reduced capacity to recover from injury also follows. We see a decreased number of satellite cells, which again divide to become new muscle cells. And we see fibrosis. We mentioned during the atrophy slide a second ago. The muscle mass is replaced by dense, regular connective tissue, which makes it less flexible. And then, of course, you've lost muscle fibers, right? So not only is it less flexible, but you've lost muscle fibers. All right, so we can't talk about muscle without talking about anabolic steroids and performance enhancing compounds. So the anabolic steroids are synthetic substances that mimic testosterone. Testosterone is a steroid, and it requires a prescription for legal use. And it stimulates the manufacture of muscle proteins. As we've said before, testosterone is a growth factor, and so it stimulates the production of, well, myofibrils in muscle. It is a so there are popular performance enhancers. I don't know what this line's supposed to mean. It's not really relevant to the rest. There are other popular performance enhancers, maybe we could say. Side effects of anabolic steroids include an increased risk of heart disease and stroke, as well as kidney damage and liver tumors. You will also see testicular atrophy and breast development in males. Now, this is a little confusing because it's not straightforward. Basically, when you take anabolic steroids, which mimic testosterone, through a negative feedback loop, the hypothalamus and the pituitary gland decide, hey, I've already got plenty of testosterone around. I'm going to stop telling the testicles to produce testosterone. And when that happens, the testicles will atrophy, they will decrease in size, and they will become less able to produce testosterone naturally. Now, you see breast development in males when you stop taking the steroids because you can't take them all the time. And if the testicles have atrophied and are not producing endogenous testosterone, then you will see breast development and other symptoms in males. The term for breast development in males, by the way, is gynecomastia. It's kind of a fun word to say. Acne, as well as high blood pressure and aggressive behavior, can result from the use of anabolic steroids. And then in women, we see the growth of facial hair and menstrual irregularities as testosterone interferes with the production of estrogen. And then, of course, we mentioned testosterone's growth factor. It also stimulates facial hair growth. All right. So, cardiac muscle cells. We're finished with skeletal. We're going to finish up with cardiac and then smooth muscle, and then we'll be done for the muscle unit. All right, so cardiac muscle, these are short branching fibers. Remember that a skeletal muscle fiber covers the length of the muscle cell, which can be long, right? In the case of the biceps brachii, that's like 8 to 12 inches, depending on the length of your arm. Now, cardiac muscle cells can have one or two nuclei. A lot of textbooks and a lot of exams out there will say this muscle is striated with one nucleus. Well, okay, it's one or two, right? They can have one or two. They don't necessarily have just one, but whatever. They can have one or two. That's important to remember, I think. And I think a lot of textbooks get that wrong. Then we have the striated appearance. That's due to the presence of the sarcomeres. Remember, the sarcomeres have the I bands and the A bands, which give us the light and dark, respectively. And so cardiac muscle is striated, just like skeletal muscle. Now, it has many mitochondria, and these mitochondria, of course, allow aerobic respiration in the cell. In fact, actually, where muscle will typically, muscle will break down glucose for energy initially. Cardiac muscle actually prefers fat. It's kind of weird, especially because, fatty foods clog your arteries, which will kill the heart. But it's true. The heart can metabolize a variety of substances, but unlike other tissues, it actually metabolizes or breaks down fat first. It prefers fat. And if you went on a zero fat diet, well, I guess you could make your own fat through lipogenesis. Yeah. All right. All right. Anyway. Yeah, many mitochondria in the cardiac muscle because that fat has to go through oxidative phosphorylation in the mitochondria to produce ATP. And then the muscle cells that we mentioned, they were short and they're branching, but they're joined together. They join the next cell over through intercalated discs. All right, it joins the ends of the cardiac muscle fibers together. And these discs contain desmosomes, which anchor them together, as well as gap junctions. Now, gap junctions, we mentioned a long time ago, those are like portals between cells, allows two cells to share cytoplasm, right? Remember the idea of a duplex. You know, a duplex is where you take a house, you cut it down the middle, you put a wall up, and then basically it's two apartments side by side in the same house, right? In a duplex, imagine if you had a window in that middle wall, right? Like your living room looked through a window into your neighbor's living room, right? They were right there next to each other. You could open that window and you could pass things through. You could pass water through. You could pass a basketball through. I mean, you might even be able to climb through, but you're not going to fit like a couch through, right? And so the gap junctions are a similar idea where we can pass certain substances through. We can pass water and ions through, but proteins are too big to get through. All right. So that's kind of key here. When we stimulate one muscle cell, remember that that causes an action potential where sodium floods into the cell and potassium floods out. We've depolarized the cell, we've flipped the polarity, and that's going to open up sodium channels down along the membrane. All right. Now, if we do that down along a cardiac myocyte, in fact, if we do we have a picture of that here? No, we don't. Okay. So if we do that, let's say it's coming from the right. Yeah, let's say it's coming from the right in this picture. We have an action potential traveling down. We have sodium channels opening up. That increases the charge of the membrane here. And that's going to cause sodium channels down the way to open up, which causes sodium to flood in, which causes sodium channels down the way to, because of the change in voltage, flip open and bring in more sodium, and so on down the line. Now we get to the end of the muscle cell where we have this intercalated disc that's going to connect this muscle cell to a muscle cell they didn't draw over here in the white. All right, we have the gap junctions between these two muscle cells. All right, and this is actually going to let, when we are depolarizing the membrane here, this is going to let sodium flood into the next cell over through the gap junctions. And that causes the next cardiac myocyte to contract, right? Causes it to depolarize because sodium's flooding in through the gap junctions. That initiates depolarization in the next cardiac myocyte, the next cardiac muscle fiber. All right, so that continues down. We see the sodium channels opening. All right, we pass down, and then at the end, sodium is going to flood. Now that it's flooded into the cell, it's going to flood through the gap junctions into the next cardiac myocyte over. All right, and so that's how we propagate the signal from one cardiac myocyte to the next. Okay, through gap junctions in the intercalated discs that join these muscle fibers together. Now, the contractions are actually started by the heart's autorhythmic pacemaker cells. We say the heart is autorhythmic, or at least the pacemaker cells are, because they generate their own rhythm of depolarization. They have basically leak channels which allow them to slowly increase the membrane voltage until it just automatically reaches threshold on its own through ion leak. And as it becomes slowly more positive and then reaches threshold, then it'll depolarize, and then it'll repolarize, and it'll wait a couple milliseconds. All right, wait for, of course, the ion concentrations to go back to normal. Waits a couple milliseconds, and then leak causes the membrane to become more positive, and then eventually it reaches threshold, and bam, it's off, depolarizing and repolarizing again. A couple milliseconds later, same thing. Leak causes it to get up to threshold, and then bam, it's off, depolarizing and repolarizing again. And so the leak channels in the pacemaker cells give them this rhythm of depolarization that's going to stimulate the rest of the heart. Okay, So in other words, the pacemaker cells of the heart set the rhythm of the heart, and they do it through these leak channels, which keep bringing the cell back to threshold and causing action potentials. All right, so the heart rate and the contraction force, though, can be influenced by the autonomic nervous system. In other words, the brain, through the autonomic nervous system, tells the heart increase your rate or decrease your rate, and it also tells it contract more forcefully or, well, no, it'll just tell it to contract more forcefully when it needs to contract more forcefully. It won't tell it ever contract less forcefully. It'll just stop telling it to contract more forcefully. Anyway, so the nervous system through the autonomic nervous system, it can influence heart rate and contraction force. I want to show you something kind of cool. In fact, actually, I've got to YouTube this, so I'm going to take that off. And I've done this before in front of a live class. You go to YouTube and you search a phrase, and uh, just weird things start coming up. There's a cool video out there, though, I want you to see. Here it is. Okay. And brace yourself. We're about to look at a real human heart, but this is anatomy and physiology, so we should be ready for that at any time. Just don't want to surprise you. Maybe you got young children who might be freaked out by seeing a heart, so I'll go ahead and give you 10 seconds before I flip it over. All right, and we're back. Let's watch this. All right, so that's a heart outside the body. It's in what's called a bypass machine, and they're keeping it alive um, so that, uh, well, the tissue can stay alive. It's waiting for transplant, basically. All right, that was pretty cool. So basically, what you're seeing here, the heart actually is stimulating itself here. The pacemaker cells are roughly up in this region. We'll cover that in the cardiac unit, but there's a conduction system that runs down through here, then down through the interventricular septum, and then back up through the walls of the heart. But the initial pacemaker cells are up here, and it is actually stimulating itself to beat. If you, as long as you supply the heart with blood that contains oxygen and nutrients such as glucose and especially fatty acids, which they are doing, right? They're supplying it with blood. You will see that the heart continues to live outside the body and continues to beat for as long as you can keep it warm, wet, and supply it with oxygen and nutrients, well, rather blood that contains oxygen and nutrients. All right, but it's actually beating on its own. It's stimulating itself. The pacemaker cells are up here. All right, and then they run down through the interventricular septum and then up through the walls of the heart. Pretty cool stuff. Beating outside the body. So there may be some truth to that Indiana Jones movie after all. Kali Ma. All right, so anything left here to say? No, we're good. Okay, we covered the picture. You've got the intercalated junctions. Remember, these are short fibers. They also can branch. They don't necessarily have to, but they can branch. That's another thing. They can have one nuclei or they can have two nuclei. There's some variation between cardiac muscle fibers or cardiac myocytes. All right, here we see one nucleus in this cell. You can see the intercalated disc. This one has desmosomes. They should all have desmosomes. These are just anchoring points, kind of like rivets that hold it together. And then the gap junctions, which are like windows or portholes between adjacent cells. Now their membrane is also called the sarcolemma, and we'll see sarcoplasm inside just like before. One other thing that's worth mentioning, I'm not sure if it's here, no in fact it's moving on to smooth muscle, is that we see, yes, you can see striation here pretty readily, that's apparent. In skeletal muscle, we had what was called the triad, where you had T tubules running down into the myofiber, and then on either side of that was a terminal cisterna. So the triad in skeletal muscle was a T tubule and then two terminal cisternae. All right. In cardiac muscle, you have what's called the dyad. And so you have T tubules, which you can kind of see here coming down. You had T tubules. And then on one side of the T tubule, you will have a terminal cisterna. Not two terminal cisternae, but one terminal cisterna. And so this structure is called a dyad, and not the triad, but the dyad in cardiac muscle. All right. And so anyway, the sarcoplasmic reticulum is present in cardiac muscle, but it's less developed in cardiac muscle. It's present, but less developed in cardiac muscle. We're going to see in smooth muscle that it is absent. There's no monoad. If you had a monoad, that would just be a T tubule by itself, right? There are no T tubules. There are no terminal cisternae in the smooth muscle. All right. Smooth muscle cells are even smaller. They are very short, unicellular, uninucleated cells. Only one nucleus ever in smooth muscle. It's never one or two. It's just one. And you find smooth muscle in the walls of hollow organs. One example is the blood vessels themselves. You also find them in the walls of the other hollow organs, such as the uterus or the bladder, the urinary bladder. Good. You find them in the bronchioles of the respiratory system that controls airflow to the alveoli. Remember, the alveoli are the air sacs in the lungs, and the bronchioles are tiny airways that conduct air to the alveoli. Gas exchange actually occurs in the alveoli, in the air sacs of the lungs. When a person has asthma, actually, inflammatory cytokines are flying around that I think one of the big ones in that case is histamine, actually. It causes constriction of the smooth muscle, which makes it harder to get air in and get it down to the air sacs. Okay, and so the treatment for that is actually a steroid. Steroids reduce the activity of the immune system typically, and we inhale one through an inhaler and like albuterol, and that's actually going to decrease the activity of the immune system there, and it's going to cause relaxation of the smooth muscle, because the immune system, by the way, is responsible for allergic reactions that cause things like anaphylactic shock and airway constriction. And this has gotten kind of big and complicated, but basically it's going to let the airways, the smooth muscle there, relax, and then that's going to cause them to dilate, and that decreases the resistance. It allows more air to flow into the air sacs. All right. We also see smooth muscle surrounding the intestines, actually the entire GI tract. Once you get about a third of the way into the esophagus, the first 1/3 is skeletal muscle, and then beyond that, it's all smooth muscle. All the way down to the anus, all the way through the esophagus, stomach, small intestine, large intestine, all the way down to the anus. It's all smooth muscle in the wall. And that smooth muscle, it contracts to push food forward or to mix it together. All right, so very important to digestion. We find smooth muscle in the walls of the ureters, which are the tubes that carry urine from the kidneys to the bladder. All right, and so when we pee, when we urinate, or micturate. Smooth muscle in the wall of the bladder contracts to squeeze it and basically push urine out. When we carry urine, though, from the bladder, I'm sorry, from the kidneys to the bladder through the ureters, that's also done through the motions of smooth muscle. They do this alternating squeezing and relaxing motion called peristalsis, which propels things along. The digestive tract also does peristalsis. Um, Peristalsis is important because astronauts in space, where there is no gravity, would not be able to urinate if not for peristalsis, right? Because peristalsis actually moves urine from the kidneys to the bladder. That rhythmic contraction of smooth muscle moves urine down to the bladder. And without that, if we were just relying on gravity to carry it down, well, in space, the astronauts they'd be out of luck. They'd be dead within hours. It would essentially be total kidney failure. You know, without smooth muscle. Very important stuff. And then the uterus, we mentioned, during parturition or childbirth, the uterus contracts, it squeezes, it pushes the baby, and then the baby, you know, exits the reproductive tract. All right, so you can see some of the locations of smooth muscle here. Um, it's very thick in the wall of the uterus, also the urinary bladder, especially. All right. So smooth muscle cells have a fusiform shape, which means they're wide at the middle and they taper at the ends. So they kind of, if you had to draw one like a cartoon, kind of look like that diamond shape. Not a gem shape, but the rhombus diamond shape. And they're smaller than skeletal muscle fibers. They're smaller than cardiac muscle fibers. They are the smallest muscle cells. The sarcolemma of the smooth muscle has varied types of calcium channels. There are gated calcium channels here, also the voltage gated calcium channels. We should say the first type there was ligand gated calcium channels. They open when they bind a substance, a ligand, as opposed to opening when the voltage across the membrane changes. But there are also voltage gated channels here. They have both types. Transverse tubules are going to be absent, as we say. There are no T tubules. The surface area is increased by structures called caveolae, which are kind of flask-like invaginations of the membrane, infoldings of the membrane, which means they're going to be wider but not as deep. And then the sarcoplasmic reticulum is pretty sparse, pretty poorly developed in smooth muscle. And then the calcium reservoir is actually going to be the extracellular fluid for smooth muscle. So here we can see those fusiform cells where, again, they have this sort of elongated shape. You can see here it's kind of like a rhombus or a diamond in the two-dimensional view, right? And then many of them are going to have these thick and thin filaments that crisscross over the cell. Instead of running parallel to one another in the form of myofibrils, we're going to see the thick filaments, the thin filaments form this crisscross mesh network that when the cell contracts, it looks like it's being squeezed by a net there. All right? Kind of looks like you're squeezing sausage with a net. All right. All right, so the cytoskeleton, is made up of intermediate filaments. There are also dense bodies, which are anchoring points within the cell, and dense plaques, where the intermediate filaments can attach to the inner sarcolemma, the inner side of the muscle cell membrane. The contractile proteins are arranged between the dense bodies and the dense plaques, and they're oriented at oblique angles, which is kind of an oxymoron because the word oblique means at an angle, right, to the longitudinal axis of the cell. All right, so the contraction here causes a twisting motion of the cell, not a shortening of the cell, but a twisting muscle of the cell. And there are no sarcomeres here and no Z discs, so no striation in smooth muscle. All right, so but like skeletal muscle, the smooth muscle filaments, they do have actin, myosin, and troponin in the thick and thin filaments. Um, but smooth muscle, unlike skeletal muscle, we see the filaments have myosin heads along their entire length, and they can form additional cross bridges. The filaments can form the latch bridge mechanism where the myosin attaches to the actin for an extended period of time without using extra ATP. Um, so we spent a lot of time talking about skeletal muscle, and basically we suck the calcium out and put it back in the sarcoplasmic reticulum to end contraction. You can imagine what it would be like if you contracted your bicep to lift your forearm and then it stayed there for 10 seconds, right? That would be inconvenient, you know? That would make lifting barbells at the gym very inconvenient. You got to wait 10 seconds to be able to relax it. And that's kind of how smooth muscle is. Smooth muscle takes longer to contract, and it's going to stay contracted longer. And so that wouldn't be very efficient for producing body movements. That wouldn't be very efficient for getting away from a tiger in the wild, right? But or for hunting prey, that wouldn't be very efficient. So skeletal muscle needs to be able to contract and relax rapidly because it does move the body. Smooth muscle does not move the body. It controls the organs and basically using this latch bridge mechanism, we can generate contraction for longer without using additional ATP. So it's an energy saving mechanism. We can generate longer contractions and use less energy through the latch bridge system. All right. We also see calmodulin here, which is a protein that binds calcium and triggers contraction. It actually takes the place of troponin. Here, calmodulin replaces troponin. The myosin light chain kinase, which is part of the myosin molecule in the thick filaments, is an enzyme that phosphorylates the myosin heads when activated by the calmodulin, by the presence of calcium. And then the myosin light chain phosphatase is an enzyme that dephosphorylates or removes the phosphate group from the myosin head. And that's required for relaxation. So you can contrast that to skeletal muscle where the presence of ATP causes relaxation. Here we actually need an enzyme to remove the phosphate group from the head. All right. All right. So the steps of contraction for smooth muscle. We see a stimulus, and this can be either ligand or voltage gated, but the ligand binding is going to open an ion channel that will then cause a depolarization that opens voltage gated channels. So either way, it's going to end up voltage gated to cause an action potential here. The calcium ions enter the sarcoplasm through the voltage gated calcium channels. Remember, calcium is found outside the cell in smooth muscle. All right, that binds to the calmodulin. The calmodulin and calcium complex binds to the myosin light chain kinase, which will phosphorylate the myosin head and activate it. Then, once the head is activated, once it's phosphorylated, we can see cross bridges form. We see the power stroke. We see pulling of actin, pulling on the thin filaments. All right, which is similar to the skeletal muscle, but it's more slow. It's not as rapid. All right, so as the thin filaments slide, it pulls on the dense bodies, and then the dense bodies are attached to intermediate filaments, which are attached to the dense plaques on the sarcolemma. And these filaments move inward, and the entire cell shortens. All right, and my baby's woken up, and she's holding my finger and won't let me pull away. She's pretty strong. Couldn't reach the key to move the slide. Anyway, relaxation of smooth muscle. We see it occurs due to a cessation of stimulus. All right, the stimulus ends basically, and calcium pumps are going to pump calcium out of the cell, out of the sarcoplasm into the extracellular fluid. And we see a dephosphorylation of myosin by the myosin light chain phosphatase enzyme. And then relaxation is going to be slow due to the latch bridge mechanism. So both contraction and relaxation are going to be slower than skeletal muscle. Skeletal muscle is very fast. All right, so smooth muscle contraction, we see a long latent period. So there's a long time between the stimulus and an increase in tension. All right, this is due to slow ATPase activity. We see a long duration. So we have slow calcium pumps. The calcium pumps that pump calcium out of the cell are going to do so slowly, which means that once the stimulus is stopped, it takes a lot longer for the cell to figure that out, right? Because calcium is still inside the cell. And you also need to dephosphorylate the myosin heads with the myosin light chain phosphatase enzyme. All right, so you get this latch bridge mechanism which holds the myofilaments in place. All right. Then we see that the slowness actually fits the functional requirements. So extended contraction means continuous tone, for instance, in the walls of blood vessels. All right, and so we don't want to be rapidly changing blood pressure in the body, and when we change the tone of the smooth muscle in the walls of the blood vessels, that is going to change the blood pressure. All right, and so by using smooth muscle in the wall, we can cause these changes slowly as opposed to rapidly. Smooth muscle is also fatigue resistant. Its energy requirements are low, as we said, compared to skeletal muscle, and it can maintain contraction without ATP through the latch bridge mechanism. Basically, we need the enzyme light chain phosphatase to remove the phosphate groups, all right, in order to cause relaxation. And so if we don't activate those enzymes, then we can just maintain contraction. There's a broad length tension curve, which basically is what we've said a couple times already, which means that it takes a long time to build up tension and a long time to release it as well. All right. All right, now smooth muscle is under the control of the autonomic nervous system. We said before that skeletal is under the control of the somatic nervous system, which means that you consciously control skeletal muscle. It is voluntary. The heart is involuntary, but it's not under the control of the nervous system at all. It is autorhythmic and self-stimulating. Okay? So the cardiac muscle fibers are going to be stimulating themselves. The nervous system is not. The nervous system tells the cardiomyocytes speed up or slow down, contract more forcefully, does not tell them contract. All right? And so compare that with the smooth muscle, on the other hand. That's going to be controlled by the autonomic nervous system. However, it can also be controlled by hormones, we should say. So one good example of that is the uterus, which is during childbirth, yes, under control of signals from the autonomic nervous system coming from the hypothalamus. All right, the brain is through the nervous system telling the uterus to contract, but also the hypothalamus is causing the pituitary gland to release the hormone oxytocin, and the hormone oxytocin will also tell the smooth muscle in the uterus to contract. All right. Okay. A lot of talking today. All right. So good. The autonomic nervous system, which is the part of the nervous system that we do not consciously control, and it typically controls the functions of internal organs. It controls smooth muscle. We see a different response to stretch as well. There's the myogenic response, which is the contraction in reaction to stretch. So there's a sort of reflexive contraction and reaction to stretch. And then there's a stress relaxation response, which is a relaxation after prolonged stretch. Basically, the muscle, smooth muscle, gets stretched and it will contract in response. And then over time, it gets stressed out from having been stretched out for so long, it's just going to give up and relax. All right, so other stimulating factors include, yes, the various hormones, low pH, low oxygen, high carbon dioxide, certain drugs, as well as pacemaker cells. All right. All right, so there are two categories of smooth muscle, multi-unit and single unit. Multi-unit smooth muscle, and this was a little confusing to me when I was a student, it's arranged in units that receive stimulation to contract individually. Multi-unit means that many of the cells are going to be acting individually. Single unit means that they all act together. So when you stimulate one muscle cell, that'll stimulate all of the nearby smooth muscle cells. All right, so multi-unit smooth muscle is found in the iris and the ciliary muscles of the eye, the erector pili muscles of the skin, the larger air passageways in the respiratory system, and the walls of the larger arteries. The degree of contraction is going to depend on the number of motor units that get activated, similar to skeletal muscle. All right. And good. Now, single unit, also known as visceral smooth muscle, is the most common type, and it's stimulated to contract in unison as the cells are linked by gap junctions. Gap junctions are absent in the multi-unit. Now, these cells form 2 or 3 sheets in the wall of a hollow organ, and they're found in the walls of the digestive, urinary, and reproductive tracts, as well as part of the respiratory tract and most blood vessels. So stimulation of the single unit smooth muscle occurs through varicosities, which are swellings of the autonomic neurons. And these varicosities contain synaptic vessels with one type of neurotransmitter, and that's either going to be acetylcholine or norepinephrine. Remember that skeletal muscle is stimulated to contract by the neurotransmitter acetylcholine, and cardiac muscle is not stimulated to contract by the nervous system. Now, it will actually respond to acetylcholine and norepinephrine. However, when it binds acetylcholine and norepinephrine coming from the nervous system, coming from synaptic vesicles, cardiac muscle will speed up or slow down. It will contract more forcefully, but it does not contract in response to those things. All right. And that's actually kind of important if you think about it. The cardiac muscle, just to go back to that for a second, needs to be contracting and relaxing all the time. All the time. And we really don't need the heart to sit around and wait for a signal from the nervous system. All right. It needs to just go ahead. It's got its orders. It needs to just go ahead and contract and relax and alternate in that pattern for the entire duration of your life. Skeletal muscle should be relaxed until it is needed. So should smooth muscle. In many cases, should be relaxed until we need to contract it. All right. So the receptors for acetylcholine and norepinephrine are scattered across the membrane of the cell, the sarcolemma, at diffuse junctions. And the numerous smooth muscle cells are stimulated simultaneously in single unit smooth muscle. The stimulation is spread from one cell to another through the gap junctions, which was similar to cardiac muscle. So contraction is synchronous. All right. And so here you can see on the left multi-unit smooth muscle. Each individual muscle cell here is being stimulated to contract individually in multi-unit smooth muscle. In the single unit smooth muscle, you see those varicosities those swellings here in the nerve fiber, and that's going to release basically a bath of neurotransmitter, either acetylcholine or norepinephrine, that will bind to receptors that are spread out across the cell surface. All right. And by just dumping these out along the surface of the cells, we can cause them all to contract in unison, especially because they're bound to each other through gap junctions that will help the signal spread from one cell to another. Like these other cells, which aren't being innervated, will be stimulated by ions traveling through the gap junctions. All right, and that's it for muscle tissue. We'll pick it up in a little bit with actual anatomical muscles. All right, thanks for tuning in.